Welcome back. Well, now that we've mastered all that stuff that we talked about in the last three videos in regards to planes, names, directions, locations, quadrants, regions, cavities, membranes and stuff like that. The next place we need to go to is the cell. Okay, now what we're going to do is we're going to have a number of sequential videos on the cell. The first one today, we're just going to be talking about the cell membrane, the cytosol, and the nucleus and the nucleolus. However, there's a lot more to the cell. The next video we'll have after this one will be involving organelles, or the small little organs, or the functional units within the cell that do specific functions. After that, we're going to be talking about the cell membrane. The cell membrane is really a very complex thing that we'll be talking about, how things get into the cell. And the cell membrane is going to be really responsible for allowing things to get in the cell that it wants to get in, and not allowing things that doesn't want the cell to get in not to get in, as well as getting out the same way. Okay, so we'll be talking about the cell membrane. We will also be talking about cell division, how the cells divide, and how the difference between mitosis and meiosis. We'll be talking about that, and then we'll also be talking about what's really another important factor is DNA and RNA. Okay, a lot of people think, well, DNA, hair color, eye color, blah blah blah, and it's really a lot more than that. Okay, it's not that truthful. What DNA does is actually has a code for how to make protein, and all these proteins run everything in the body. So anyway, we'll be talking about that in that order, and then we'll be heading on to other things. But let's just talk a lot about the cell for a while, because that's really an important part. Well, where do we start? Well, let's start at the beginning, simply because, if we look at myself, and well, probably don't want to do that, but if you look at your body, okay, what you're going to find is there are so many different types of cells. You have more than 200 different types of cells in your body. You have liver cells, you have brain cells, you have smooth muscle cells, you have skeletal muscle cells, you have cardiac muscle cells, you have cells that are on the inside lining of the intestine. You have kidney cells, all these different cells, multiple, many different types of cells, connective tissue, tendon cells, ligament cells, cartilage cells, bone cells, blood cells. All these are different types of cells. And the thing about, and we'll be talking about most of those during the course of the semester, but what we need to recognize about the cell is the basic living unit of the body. It's both living as well as has structure. It's the smallest thing in the body that's alive. Okay, there are smaller things in the body that are alive, but they're really not part inherently of the body. Bacteria are significantly smaller than cells, but they're really not really what we're talking about here. But so the cells are the basic units of the body, which are both living and structural, holding things together as well as having some specific function. Like I say, they're a little bit of an island to themselves, because what happens is they're enclosed by a cell membrane. Okay, and we talked about that. We'll be talking a little bit about it today, but we'll be talking a lot about it in an upcoming video. And the cell membrane is really important because it really controls what gets in and what gets out. It's the master bouncer of the body, what lets in and what it doesn't. Cells are capable of dividing, at least most of them, okay, but not all of them. Typical example is a mature red blood cell will not divide. Once it gets past a certain point in the bone marrow, red blood cells, white blood cells, they can't divide anymore. They follow their route, and that's what it is until their demise. Red blood cells are actually made to last about 120 days, and basically all they are is they're an envelope, which is that cell membrane, and loaded with a whole lot of what we call hemoglobin, a protein which is able to carry oxygen, but that's it. And they don't really do very much of anything else. And so as a result, they have a certain lifespan. Eventually what happens is they don't divide, but they just go to some red blood cell vision in the sky somewhere, and they're done. Okay. Also, cells have different roles. Hepatocytes, which are liver cells, have multiple different things that they could do, and they're different from what skeletal muscle do. Skeletal muscle is made to be able to contract. Cardiac muscle is made to contract, but they're a little different. Skeletal muscle and cardiac muscle have differences. Smooth muscle that lines the inside of my intestines, inside of my blood vessels to make them get larger and smaller inside some of the airways of my lungs and stuff like that, they still have a different function. And what they do is they still contract, but in a different way. Brain cells, they conduct impulses. You know, kidney cells, they are involved in making and processing our blood to be able to make urine. So all these cells have different functions. Tendon holds muscle to bone, ligament holds bone to bone. So all these cells have different roles. Okay, and then, like I mentioned before, the cells are a complex organism. Okay, basically we have the outside, which is that plasma, or the cell membrane, which is on the outside, and this is loaded on the inside with a big fluidy filled bag, and this fluid filled center is basically called the cytosol. Okay, and we'll talk a little about that today. Well, floating around inside the cytosol are these smaller little structures that are called organelles that do different things, and we'll talk about those as time goes on. And we also have what are called inclusions. So we'll be getting all these. Today we're just going to be talking about the plasma membrane, the cytosol, and the nucleus and the nucleolus. Okay. Oh, there's a selfie. So everybody stop, take a selfie. Here you go. You're ready to go. So let's talk about the cell. Okay. And the three main parts of the cell are the plasma membrane. Okay. So if I take a picture and take this image right here, and I look, you can see this membrane that comes around the outside here, and that's my plasma membrane. Okay, it's sort of a complex structure, and we'll talk a little bit more about that, how it's made up, and the structure of it really is significant. Okay, it's flexible, so there is a little bit of a bend to the shape of the cells. They usually have some characteristic shape. When we talk about tissues, you'll see how these characteristic shapes are. Okay, what it does is it separates the internal environment in here from out here, from what's out in the outside. And again, some things on the outside need to get in, some things on the inside need to get out, but there are things that on the outside you don't want in, and the things inside you don't want to go out. Okay, so basically what this plasma membrane is, it's a selective barrier. A selective barrier. It lets things in that it wants to, and other things it doesn't want, it doesn't let in. Okay, inside that plasma membrane, I have what's called the cytoplasm. Okay, and the cytoplasm, or the cytosol, is all this bluish stuff that's all inside. And basically, it's everything between the plasma membrane and then what's this thing in here, which is going to be the nucleus. So it's everything inside there. Now, within that cytosol, that liquid portion that's inside the cell membrane, is basically a number of suspended particles. If you look here, let's see, what do we see here? We see a bunch of pipes here coming up here. You see a bean over here. They're not really a bean or anything. They're not really pipes. You see these little, like, folded sheets or something like. No, it's not like that. What happens, these are things that are called organelles. Okay, and organelles, or little organs, have various functions. Typical example, ribosomes. Ribosomes are places that make protein. Okay, mitochondria are these little beans here, are basically things that produce energy. The Golgi complex, like this area in here, is basically to try to package, organize, and distribute proteins and other materials throughout the cell. So and then lysosomes. Here's a little lysosome right there. There's a little lysosome right there. And basically these are things that have like digestive enzymes in. So when things do get inside and they want to get rid of them, they actually merge them with the lysosomes. The lysosomes will sort of dissolve them so they could get rid of and be extruded to the outside, like this little, like that little puppy out there. So we'll talk more about those, about the organelles, in the follow-up lecture to this. Okay. The final thing that we have here is the nucleus. Okay, and the nucleus is this puppy in here. Okay, it doesn't look like it's like a clam that's open. It's a round circle. Okay, and what's in the nucleus? Only something small called DNA. DNA is in there. We'll talk a lot about DNA over the next couple of weeks. What really it does, and what happens is inside each nucleus of each cell, okay, of the body, and like I probably could say with very significant certainty that most cells have a nucleus, but we're going to have a good old friend like we talked about before, the red blood cell. Red blood cell, no nucleus. Okay, that was actually gotten rid of early so they could just pack it all with that hemoglobin. So no nucleus in the red blood cell. But inside that nucleus, we have what are called chromosomes. And this is where the DNA is. We'll talk a little bit more about it at the very end of today. We'll talk a little about what that chromosome is. Basically, the chromosome is a winded strands and all twisted around of DNA. And what we have is we have 23 pairs. That should be a 3 pairs of chromosomes, and the 23rd pair is a sex chromosome, which is either an XX or a female, or an X male would be an X Y. Okay, we'll talk a little bit more about that. What's on these chromosomes are little small sections on the chromosomes, and those little sections of the chromosomes are called genes. And the genes actually have the code for making every protein, everything I need to know. Everything I need to make my body are on these genes on the chromosomes inside the nucleus. Okay, the nucleolus is another little part inside the nucleus, and that nucleolus is an area which is exceptionally important in regards to the making of proteins. Okay, it's where things are starting to become assembled in that area of the nucleolus, which we'll talk about a little bit later. Okay, but the chromosomes are look like little X's inside there, little X's inside there, and there's 23 pairs of them. Okay, and we'll talk about those. I'll show you a map of them a little bit later. Okay, so let's talk more about that plasma membrane. And the biggest thing about the plasma membrane, again, like I mentioned, it's selective permeability. It lets in only what it wants to get in. Okay, and this is an important factor. In other words, a cell has to be very selective as to what it needs and what it wants, and the cell membrane is really responsible for doing that. Now, things will get in and out get in and out through the plasma membrane in a number of different ways, and that's going to baffle your mind. You're going to be sitting there, you're going to be staying up late at night trying to remember all the different ways things get in and out. But there are, yeah, it's just one of those things you have to think about. Okay, but what happens is the plasma membrane needs to be functional to keep the cell viable and intact. Once a cell loses its plasma membrane stability, the cell will die, because it can no longer control what gets in and what gets out, and everything gets in and everything gets out, and it becomes a real mess, and the cell will die. Okay, so basically, in some ways, it's a way of communication between the inside and the outside by saying, what do I need, and then bring it to me, and I'll make sure I can get it in. Okay, so that's it. Also, another thing, and we'll be talking about this a little bit later. Okay, not today, but a little bit later in multiple areas, it creates what's called an electrochemical gradient. Okay, what electrochemical gradient is, what we know is things are either could be a positive or a negative charge. Okay, positive or negative charge. What happens is that depends upon the component inside the cell and components outside the cell. Let me just give you a little idea. Let me just draw a cell right here. Now that's a cell membrane, supposed to be. Okay, take it from me that that's what that's supposed to be. The inside of a cell is more what we call electronegative, electronegative. Okay, and the reason why it's electronegative is because a lot of the stuff inside the cell are large structures called proteins, and proteins are frequently electronegative. So the inside the cell has a relatively electronegative charge. The outside the cell is a relatively electropositive, and the reason why is because the cell, while it has a soft fluidy center, it's surrounded by what we call ECF, or extracellular fluid. Extracellular fluid. And one of the big components of this extracellular fluid is a cation called sodium. So that we have a lot of sodium on the outside, and the sodium on the outside gives the outside electropositive charge. So the inside is a little bit more electronegative, and the outside electropositive. I want you to remember that for now, but the significance of this is going to come up a lot more when we talk about cardiac muscle contraction, about nerve conduction, about skeletal muscle contraction, and other things. So we'll be talking a lot more about this as time goes on. But just remember, the outside of the cell is a little bit more electropositive, and the inside of the cell is more electronegative. Also inside the cell is another cation called potassium. Okay, so we have potassium in, sodium out. Now, potassium is also what's called a cation. Okay, if you hear me say the word cation, C-A-T-I-O-N, it's an ion with a positive charge. With a negative charge, it's called an anion. A-N-I-O-N, and that has a negative charge. Okay, so potassium is a cation inside and sodium is a cation outside, but we have a lot more of the electronegativity on the inside because of a lot of proteins and stuff like that. But this is going to be important, so I want you to remember the sodium on the outside, and I also want you to remember the potassium on the inside, because sometimes what happens is things will actually cause the sodium to rush in, and what actually what tries to balance that out initially is potassium goes out. So sodium rushes in, potassium rushes out. But we'll talk more about that later on. Don't bother your mind Not with that. So I think I'd like you to know about cations and anions. So cations are positively charged ions, anions are negatively charged ions. Okay, so let's go on from there. Oops, you can't even see my good work up here. Get rid of all these negatives and see what does it say here. Why did the protein cross the cell membrane? Don't know why. To get to the other side. That's stupid. All that erasing for that? Okay, come on, give me a break. Okay, let's talk about that plasma membrane, because this is really critical. This is really critical. First of all, plasma membranes are basically a lipid bilayer. Lipid means fat. Okay, and there are three types of fats that we have in this, three types of lipids, okay? One's called a phospholipid, which is the majority of it. The phospholipid's the majority. There's also a little bit of cholesterol, a little bit of glycolipids. Here's what I want you to remember. Majority of it's phospholipids. So I'll explain what that phospholipid is in a little bit, okay? It also has proteins in there, and these proteins are mostly what I call glycoproteins. It's protein with a bit of a sugar base on it as well, and they have different functions. If you look here, let's look at this right here. You see this hot dog here? Guess what? That's not really a hot dog. That's a protein through the wall of the cell membrane that will allow things to go in. Same thing with all these little curlicues here and the little firecrackers over here and stuff like that. Basically, these are proteins that are embedded or floating in the membrane. And most of these proteins are there to either pick something up and carry it across the membrane or whatever, or sometimes we have what are called receptors. Interesting, because if I took somebody's kidney and stuck it in my body, my body would reject it. Why? Because on the surface of all those cells, there are proteins that say, this is me and me only. And as a result, if you stick it in my body looks at that and says, hey, that's not you. That's him over there. Therefore, what it's going to do is going to cause an immune reaction. It's going to destroy that kidney. Okay. So some of these things would be receptors. Some of them will be cell markers. Some of them are transporter proteins and things like that. And like I say, these proteins sort of float like icebergs in a sea of lipid. You'll see that almost every A&P textbook. Proteins floating like icebergs in a sea of lipid, sort of cornbally, but that sort of gives you an idea. And you can see all these little proteins that are floating around there. Some of these proteins are called transmembrane. Transmembrane, like this one right here, it goes all the way across. This one goes all the way across. Some of them are peripheral. There's a peripheral one right there. Some of them are integral. They're basically only inside the membrane. Okay, and they act in a bunch of different ways. Okay, so what we see with the phospholipids. Okay, phospholipids are basically. Whoops, let me get out of there for a second. Phospholipids are made up of two fatty acids, okay, which are long-chain carbon and hydrogen molecules. And these areas are called hydrophobic. And these areas right here, these are the area which are these long-chain fatty acids, okay, the hydrophobic area, okay. And hydrophobic means that they don't like water. Now think about this. Lipids are what? And this makes sense. And this is easy to remember. Lipids are what? They're fats. Take some oil, which is a fat, and drop it in, and take a little, and pour some into a glass of water. What happens? They separate. Why? Because the fats don't like water. It separates, which basically then, therefore, this whole inner side right here is good, because it actually, the outside of the cell is mostly what? What's the outside of the cell? Did I hear it? Mostly water. The inside of the cell is mostly what? Did I hear it? Mostly water. So actually this lipid layer in the middle that's basically up the center right here, this area in here, is basically sort of like helping to prevent the water from getting through. So it's like, putting oil in water, and that's what happens, okay? So those are the two long tails. You can see these tails, how they are over here, how they sort of like, let me just get rid of that, how they sort of like, they face each other, okay? Okay, so on the outside, this blue area, let me just do a different color. Let's do it green here. Okay, so teal color. This area right here is called a glycerol head. Okay, and it has a phosphate group, and this part likes water. Wow, a match made in heaven. Why? Because if I look at my glycerol layer here, and if I look at my, whoops, that's a bad choice. Okay, let's do the glycerol layer in this right here, glycerol layer right here, and let's do the fat layer in here, coming down this way and up this way. What happens outside is water, H2O, H2O. This part here, the blue, which is the glycerol area in here, loves the water, so it can exist very easily with it. However, that hydrophobic area in the center doesn't, creates a little bit of a barrier. Makes sense. I think if you just think about it, it makes all the sense in the world, okay? And so what the cell membrane is overall is like a wall, okay? It's a wall that's able to, keep things in and keep things out and protect things that are inside and make sure everything is working and everything is, there's a little separation of power between that. So there's my wall, there's my cell membrane, and that's what we see, okay? Now, this lipid bilayer is also called amphipathic, okay, which means it has a polar and a non-polar part. Polar and non-polar, okay? Polar versus non-polar. What happens is the head portion is polar, which means it's a hydrophilic. It likes the water. The tail portion is non-polar. So this in here is non-polar. This area on the outside is polar, okay? So the non-polar area is hydrophobic inside, and the outside, which is polar, is hydrophilic. It likes the water. Okay, so that's a little bit about that. There's a little bit of description on the bottom: polar versus non-polar. In a if you've taken chemistry, you know about polar and non-polar bonds. Okay, in a non-polar bond, what happens? I have two charged. Let me get my picture back up here again. In a non-polar chemical bond, okay, I have a charged particle here and a charged particle here. This one's usually a positive, this one's usually a negative. But what they do is they share their electrons to make themselves stable as they bind together. So therefore, even though this one's positive, this one's negative, they balance themselves out and there's no charge to the molecule, okay? And that's called nonpolar. That's called nonpolar. And again, what we find is the center portion in here is nonpolar. On the other hand, polar molecules basically have a positive charge and a negative charge, but what happens is one of the molecules or the ions that has too many electrons will donate. We talked about this in a previous discussion. So as a result, what happens is part of the instead of neutralizing themselves out, one half becomes very charged, okay? So therefore it becomes polar, okay? So non-polar, no charge, the electrons bond to each other, they cancel themselves out. Where in polar molecules, what happens is part of the molecule has a positive charge and part of the molecule has a negative charge, okay? And that's what we see in the surface. Again, looking at that lipid bilayer, the heads point outwards to the glycerol head that we see on the outside. Hydrophilic, they like water. They're basically polar. They work really well. Everything is great. They bond well with the water. So the environment, the water on the outside is okay with them, and the water on the inside is okay with them. Everything is just hunky-dory. On the inside, though, we have that hydrophobic non-polar area on the inside, which basically is that separating layer. So we have to live with our environment by having an outer shell that's compatible with the environment, water on the outside, water on the inside, but I want to separate it and make sure that membrane is limiting what comes in by that center hydrophobic area of mostly the lipid tails. Okay. Now, that's a little about the cell membrane. You think that's all? No. We're going to have a lot more, okay? And we'll talk more about how things get in and out of the cell membrane, which is very complex. So get ready. You might want to order a seatbelt just to, keep yourself stable in front of the computer when you're watching, okay? Let's talk about the cytosol. Cytosol is about 75 to 90% water. Most of it's water. It's semi-fluid, and semi-fluid because what happens is there's a lot of stuff floating around in it, okay? And what's floating around in that? Again, these inclusions, which are chunks of materials that are floating around, like the proteins are floating around inside there, dissolved solutes, potassium is floating around there, other types of ions are floating around in there, large organic molecules, and they're called colloids because they're suspended. So it creates almost like a gel almost, okay? And some of these things are basically what we'll see that are floating inside there are really common things such as ions, which are charged particles. Glucose is in there. Glucose is a very important molecule because it's a clean burning fuel like we talked about, okay? Amino acids, critical. How do I make proteins? What are proteins? They're chunks of amino acid. We take amino acids, put them together. We make a peptide, which are amino acids that are held together, and then they become polypeptides when I take multiple peptides and bring them together into a very large molecule called a protein. Most proteins are really huge molecules, by the way. Okay, we have fatty acids. We have other proteins floating around there, lipids, ATP, which is an energy form. We'll talk a little about that, and a lot of waste products that the cell has to get rid of. So these are just things that are floating around in the fluid portion of the cytosol. But what else is floating around in the fluid portion? The organelles. Those organelles are those little organs like we'll talk about in our next little video. Okay, so what happens in the cytosol? What really happens in the cytosol? A lot of chemical reactions, a lot of things. You know, the cell needs to function, and even though we have all these organelles in there, the organelles are always working. We're making energy, we're making new materials, we're making proteins, we're doing all kinds of stuff inside the cell. So as a result, the cell needs a number of chemical reactions to occur for allow these processes to occur. A typical example is glycolysis. Okay, so how do you use glucose? Well, glucose goes through a very complicated transformation, because glucose by itself means nothing, but then the glucose is broken down into smaller chunks, and by doing that it actually goes through a series of chemical events that allows energy to be produced. Okay, and that's called glycolysis. Glyco means sugar, lysis means to break things up. So that happens in the cytosol. Okay, a number of other things happen in the cytosol. So it's a site of a lot of the production of what happens inside the cell. Okay, isn't that cytosol? Now, the organelles, okay, again, are these specialized structures, materials, or little organs within the cell, and they're involved with a lot of things: cellular growth, maintaining the daily functions of a cell. They're also involved in making preparations for reproducing the cell or cell division. Okay, and all these organelles have their own sets of enzymes, and if you know anything about enzymes, they're substances produced by a living organism which act as a catalyst or make things happen. They bring about a specific biochemical reaction. So there's tons and tons and tons of enzymes, and these enzymes, guess what they are? Oh, that's right, they're proteins. We have to make those. Okay, so each of these organelles, though, has its own special functional compartment. So in other words, when you look at it under a cell, under a microscope, particularly under exceptionally high power, what you'll actually see is you'll see little pieces, parts inside the cell, and these little pieces, parts are parts of these organelles, such as mitochondria and all kinds of stuff like that. Okay, so each has their own specific functional compartment, which is very has characteristic appearance. They look like, you know, what they should be. Okay, again, and the number of these organelles vary with the type of cell. Typical example is if I look at muscle cell, skeletal muscle cell. Skeletal muscle cell, guess what it needs? It needs a lot of energy. So therefore, what makes energy? These things called mitochondria. Therefore, skeletal muscle has lots of mitochondria within that cell, a lot of those organelles of mitochondria inside the cell to make a lot of energy. Okay, so basically things will change according to which cell, according to what their purpose is. Okay, if you look at things such as white blood cells are basically cells that like to destroy things. So therefore, they have a lot of lysosomes, or these organelles that will actually have a lot of digestive enzymes to break things down. One way is when bacteria will be engulfed by a white blood cell, gets inside, and then the bacteria eventually gets fused to a little membrane around the lysosome, and the lysosome releases sort of enzymes, and as it brings it sort of like it fuses with it, and it destroys and it causes destruction of those bacteria. Now with all the COVID stuff going on, hopefully maybe 10 years down the road, if everybody's ever looking at these videos, they'll say, COVID what? Anyway, what happens? COVID is a virus. A virus is not living organism. Bacteria are, viruses aren't. Viruses are actually pieces of DNA or RNA. If it's an RNA, it's called a retrovirus, and the other viruses are DNA viruses. And what happens is this virus has a chunk of DNA. What does that chunk have on it? Well, it has genes, just like genes like we have. These genes code for various things, such as proteins and different other processes that are going to be done because of the proteins that it codes for. So what happens is in a virus, a virus is usually encapsulated in a capsule. and it gets inside the body. The body engulfs it, okay, and what happens, it gets inside this capsule around the virus will disintegrate. The virus gets release the kraken, and all of a sudden what happens is now that virus goes to the nucleus, gets incorporated in the nucleus in our own DNA, and therefore it has a code for making these abnormal proteins which cause disease or which cause other symptoms and stuff like that. So that's a little bit about viruses. That's why you can't kill a virus. You can control them in certain ways, but you can't kill them. People with chickenpox as kids, they're much more, they run a risk later on in life of having shingles, which is called herpes zoster, which is basically these chickenpox virus particles stay, and they like a certain area, like along nerve roots. And what happens is sometime when your immune system's a little bit down, bam, they come out and they just go crazy, and the next thing you know, they're making all kinds of problems with all kinds of look like chickenpox and look like a like a little stripe, usually along your back, sometimes on your face and stuff like that. When people have cold sores are basically herpes simplex type 1. Guess why? Guess why people keep on getting cold sores if they've got one? Because they can't get rid of the virus. You can control it, you could decrease the time it's there, but you can't kill it, okay? So as a result, you might control it. It says, okay, fine. You know, I'll go back and go to bed for a while. It goes back and lays down for a while until it's time for it to get up again, and boom, it comes up again, usually around the time of stress, like finals, exam times, first date, you know, stuff like that, you know. So then all of a sudden your immune system gets a little bit stressed in those times of other types of physical or psychological stress, and the viruses go crazy. But let's get back to these organelles, okay? So also these organelles, again, they all have their own specific functional compartment. That type and number vary with the cells, and they cooperate to maintain homeostasis. Now, we talked about homeostasis. The body likes to be the same all the time. So to be able to keep the environment within the cell constant, these organelles work overtime. Okay, now when we look at the organelles, you know, these are a whole lot of organelles. You know, we have the nucleus, we have the ribosomes for making protein, endoplasmic reticulum, which is basically where a lot of the ribosomes are. They're like channels inside the cell to let things move around. A Golgi complex that packages and processes all these proteins, the lysosomes for destruction of things. Peroxisomes also destroy things, just like the lysosomes, with peroxides and stuff like that. Mitochondria, again, making energy. Cytoskeleton sort of gives the cell some structure. Flagella and cilia, these are not common on all cells, okay? Most cells don't have that, but these are either like little hairs on the surface of the cell or like a whip-like tail. Spermatozoa swim. Why? Because they have a flagella that makes them swim. The inside of your respiratory tract has cilia, and the cilia trap little particles that we might breathe in, okay? We also have centrosomes and centrioles, which is basically something we'll talk more about when we talk about cell division down the road a little bit, okay? Okay, let's see. Everyone do what I say. Haha, no. You guys be quiet. I have so much work to do at the nucleus, okay? Everything I touch, I destroy. Oh, that's a lysosome. I feel quite fulfilled, which is basically a vacuole. Vacuoles are basically vats that store a lot of garbage that they want to get rid of, okay? I need a break, okay? It looks like a Golgi, stuff like that. Mitochondria, you know, they're basically going to be making a lot of energy up in there. Okay. Stuff. There we go. Anyway, let's talk about the nucleus and round this discussion out. The nucleus is the control center of the cell. Okay. Again, most cells have a single nucleus, with the exception of some. Red blood cells don't have a nucleus, like I talked about before. To maximize the amount of carrying capacity they have for hemoglobin, which is carrying oxygen, which is the sole purpose of a red blood cell, basically they throw them out. The red blood cells initially had a nucleus, but as they mature and develop inside the bone marrow, they actually start to break up the nucleus. The nucleus has gotten rid of it. It's just destroyed and eliminated from the cell. Sometimes when people have a hemorrhage, we'll actually see you'll see red blood cells still with a nucleus, which means the bone marrow is putting them out before they should. They don't really work as well, but they're putting them out too early, too young. They're called reticulocytes, but we don't have to talk about that. Also, muscle cells frequently have more than one nucleus, you know, because the muscle cells, to be truthful with you, are really, really long. They're long spindles, okay? What happens? What's in this nucleus? They contain our hereditary information, okay? all of my hereditary information is in the nucleus. Okay, we have 23 pairs of nucleus, or 23 pairs of chromosomes. Okay, and on the chromosomes are small little areas which have specific recipes, specific codes, and these little recipes or codes are called genes. Now, this is an interesting thing about chromosomes. The majority of the chromosomal material, you know what it means? Close your eyes. Just go ahead. Humor me. Close your eyes. What do you see? Nothing. Most of it doesn't mean anything, and some people actually think it's basically areas where old dead viruses, well, excuse me, I should have said dead because they're really not dead because they're never alive in the first place, but old viruses that don't work very much anymore are parts of these old chromosomes possibly. And then if you're really spooky, woo, and think about, people say, oh, that's just alien DNA, it doesn't work, but we're not going to go there, okay? Although, because what'll happen is they'll probably call the dean, and the dean will have me carried away in a straitjacket. So it has nothing to do with aliens, okay? So all this hereditary information is on genes on the chromosomes, okay? And what the genes do, okay? The genes are basically there that actually have a code for making protein. For making protein, it should be an N. Didn't come out very good. Protein. Okay, so that's what's on the genes. Code for making proteins. Okay, so what happens is that's really all that the chromosomes do. Okay, they have the code for making all these proteins. So basically, what they do is these proteins are used. Excuse me. These proteins are used to carry on a lot of the daily cell activities. Okay, things that are going on every day. Okay, inside the nucleus we also have a nucleolus, and the nucleolus is there for a purpose. It's for the production of what we call ribosomes. And what ribosomes are is they are areas where protein is made. Protein is made on the ribosomes, and these ribosomes are made and contained inside the nucleolus. Okay, so basically these ribosomes are it's a macromolecular machine, serves as a biological site of protein synthesis in the ribosomes. Okay, and what do the ribosomes do? They make protein. How they make protein? Well, if you've listened before and you didn't fall asleep earlier on, you realize proteins are linking amino acids in the right order, in the right sequence, in the right length of the chain together. Okay, and we'll talk about how that happens when we talk about what DNA does. So it takes these amino acids and link them together, specified by what we call messenger RNA. And messenger RNA says how to make these proteins. We'll talk about messenger RNA. We'll talk about transfer RNA. We'll talk about ribosomal RNA down the road, which is a little bit different from DNA. DNA is a double-stranded material. RNA is a single-stranded material. We'll talk about that a little bit later. And what does that say up there? Let's get rid of that. Okay, here's the nucleus. Okay, nucleus. So this is insightful. With great powers comes great responsibility, and there's a piece of DNA that's sitting inside the nucleus. Okay, this is looking. It's just a photo, a micrograph of a nucleus on an electron microscope, and you can see the nucleus. Okay, and let me just show you and try to get you. I'll explain this. This right here outside is called the nuclear envelope. Okay. It's around the outside, and it's basically like the cell membrane of a cell, but it's around the nucleus. It separates the nucleus because, again, the nucleus needs quiet. It needs to be by itself. I don't like to work in big groups, you know. It needs some quiet. And then inside the nucleus, I have this mass of material here, which is basically called the nucleolus. Okay, so we have this nuclear envelope, which is basically a double-layer membrane that separates from the nucleus from the cytoplasm. Okay, it's a lipid bilayer, just like we saw on the cell membrane. Okay, the outer portion is actually continuous with what we call the rough endoplasmic reticulum. What happens is, again, in that nucleolus, I'm making ribosomes. And the ribosomes are for making protein. What happens is we have what's called the endoplasmic reticulum. If you look down here, see where it looks sort of all these lines, all these look like little channels here? See some here right there? That's called the endoplasmic reticulum. And basically they're consistent with the nuclear envelope, okay? And so what happens is as the proteins are being produced, and we'll talk about that process down the road, they could get pushed out into ribosomes in these little folds, which is called the endoplasmic reticulum. We'll talk about that later on. If the endoplasmic reticulum has ribosomes on it, it's called the rough endoplasmic reticulum, because when they look underneath the microscope, it looks rough because it has these little ribosomes, little balls all over it. If there's endoplasmic reticulum just for passageway and nothing else that doesn't have ribosomes, it's called, guess what? Smooth endoplasmic reticulum, because it doesn't have those little balls, okay? Again, those nuclear pores are basically little openings. In a couple of places you could see them. Let's see, oh, there's a good one right there. If you look right in there, there's a little nuclear pore. I think there's a little nuclear pore that you see right there. There's a nuclear pore right there. There's a nuclear pore right there. There's a nuclear pore like there. And it allows a little bit of a communication between inside and outside, okay, through that nuclear pore. And that nuclear, and the nucleolus, like we talked about, is basically a cluster of DNA and RNA, which basically is a cytosynthesis of what we call rRNA. rRNA stands for ribosomal RNA, okay? And this basically is what the proteins are going to be made upon, and that'll make sense to you later on down the road, okay? So think about that later on down the road. So let's look at that chromosome, okay? And this is what's inside the nucleus. The nucleus has these chromosomes, okay? And what the chromosomes are is it's basically this DNA double helix, okay? If you look like it's like a spiral staircase, okay? And we'll talk a lot about the structure of it. And basically this outer shell that comes around here is basically a sugar. And there's two sugars that's why they call it deoxyribo, because deoxyribo is two sugars, okay? D is 2. Anyway, what happens is, and then we have what are called nucleotides, nucleic bases that come across the inside that link this together. We'll talk about that later on, okay? So these are the hereditary units, or they contain the hereditary units, and the hereditary units on the chromosomes are called genes. And again, if I look at this big pot of ramen down here, okay? If you look at that big pot of ramen, basically only small little sections of those. Well, actually the genes could be quite long, but they could be really small as well. Maybe only a couple sequences long. But a lot of this means absolutely nothing. A lot of that twisting and turning means nothing. They all have this X-type shape. They come together in a centromere right in the middle, almost like some of them are big, some of them are small and stuff like that. We have 23 pairs, okay? We have 23 pairs of chromosomes, okay? And the 23rd pair is called the sex chromosome, like I mentioned before. What happens if it's XX, it's female. If it's XY, it's male. We'll talk about that later on down the road. And what this chromosome is, it's all this DNA that's wound together in a big long strand. It's all spaghettied up, you know, or like ramened up inside there, and like big strands that come down, and big strands are wound up and stuff like that. Okay, I'm having fun. They're all wound up in there, and basically that's what it is. It's a long strand that's all coiled together. A long strand of DNA coiled with several proteins in there. Okay. What happens if we look at a cell? If we look at the nucleus of a cell, what happens? We see a material called chromatin. Okay. When you look at it in the microscope, okay, and I'll show you some microscopic, some tissues and some cellular pictures down the road in a microscope lab, even though we don't have microscopes. Okay. I'll show you some pictures of the nucleus. You'll be able to look at the nucleus. And the nucleus, you'll see these clumpy areas. And the clumpy areas are complexes of DNA, protein, and some RNA that you see within the nucleus of a cell when you look at it. It looks like granular, clumpy, diffuse mass. It's inside the nucleus. And basically that's just DNA and other material that's inside the nucleus of the cell. One other word I think you should know right here is called genome. Genome is the total genetic information of a cell. Now, a number of years ago, scientists said, hey, you know what? Wouldn't it be nice if we knew what gene was where? Why? Because if a gene goes bad, or if a gene mutates, it makes a bad protein, okay? What do genes do? They have a code for making protein. So if a gene goes bad, the protein that it makes is also going to be bad. Garbage in, garbage out. If the protein is bad, what happens is that it may cause disease. You know, typical. You know, there's a typical example. There's a muscular dystrophy. Now, there are different types of muscular dystrophies, but the one that's probably the most devastating is called Duchenne's muscular dystrophy. Duchenne's muscular dystrophy, by just for your information, is in males only. And it's in males only because what happens is the abnormal gene is on the Y chromosome, which is only in males. Okay, females would be XX, males for XY. So somewhere on the Y chromosome is an abnormal, or is a genetic mutation for making an abnormal protein, and that protein is responsible for making muscle. Okay, different types of, or I can't say muscle, but making a certain protein that's used in muscle to make muscle strong. And as a result, what happens is that's not there, because along the way there's been a mutation of that gene. Cystic fibrosis is another genetic disorder. Okay, that's on the 7th chromosome in a certain spot. We'll talk about how we could actually map them. So people said, if we know where the genes are on a chromosome, we might be able to do something to determine where a problem might be in a disease, or down the road, even things like gene splicing or gene modification, which may help people to maybe cure certain genetic type things, which is actually still way off in the future. Okay, but anyway, because you still have cells. So anyway, so it's that they're old ones with the bad genes. But anyway, we'll talk a lot more about this. But genome is basically the total genetic information that we find of a cell. Okay, so that's a little about chromosomes. This is just looking at that. Here's a chromosome. There's a chromosome sitting right there. And if we look, basically, again, it's all this DNA that's coiled around and held together. Here's the DNA, and it's coiled around, held together with various proteins and stuff like that, and it creates this chromosome. Now, this over here is a chromosomal map. Okay, it's a chromosomal map, and basically what they could do is they could take the nucleus of a cell and extract the chromosomes, and they could map them to look for certain abnormalities and things like that. So up here would be chromosomes 1. Here's the pair 1, pair 2, pair 3, 3, 4, 5, 6, 7. Let me look at this one right here. This one right here is the 21st chromosome right here, and a lot of times, if you've heard something called Down syndrome, that's not P, that's a Down syndrome. Down syndrome is basically a 21, what's called trisomy, which means instead of having 2, it doesn't, has 3. Has 3 chromosomes, so there'll be an extra one in here besides that. So they can actually determine a lot. When they do looking at a fetus before it's born, they do an amniocentesis, stick a needle inside the amniotic sac and suck out some fluid. And when they do that, some fetal cells will be floating around there like a snow globe inside the amniotic sac. And then what they do is they spin them down in a centrifuge, take those cells out, and extract the DNA so they could look for genetic abnormalities or DNA type problems. And what we'll find out later on is each little part in the DNA, each little part in the chromosomes, okay, with a particular gene could be mapped. So they know, they did the genome project, which basically the idea was to go and map everything they could, and they did a really great job. So we know a lot about what's where on which chromosome, so that might help us down the road in regards to sort of helping certain diseases, or at least right now identifying various conditions. So what have we talked about so far? We've talked a little about the outer portion. We talked about the cell membrane, but there's a lot more to come, believe me. We just hit the surface in regards to cell membrane, because remember all those little floatines, like floatines. That's good. I'll have to remember that word, floatines. Floatines are proteins floating, in that lipid bilayer. So that's in that sea of lipid. Okay, icebergs, protein icebergs floating in a sea of lipid. Sort of. I think it reminds me of the Titanic. But anyway, if you there's a lot more we're going to talk about in regards to that, how things get in, how things get out, and what a lot of these proteins are for. Okay, we'll talk a lot more about the cytosol, but not as much as the cell membrane, and we will talk a lot about the nucleus, particularly in regards to DNA, how the DNA works to be able to produce the protein from RNA. Okay, and that's it's sort of interesting, because students get, oh my gosh, I got a headache, I'm so I can't understand, oh, it's so hot, tough, oh my gosh, I can't think anymore. What happens when you look at it, it's actually quite simple and quite straightforward. It's beautiful the way it's set up. So anyway, we'll talk more about that in a couple weeks. So anyway, that's what we talked about today. Hopefully you learned something, and hopefully this will set a nice little foundation for what's going to come in our next discussion. Okay, for now, be good, stay safe, be healthy. Talk to you.